screen share.
Yeah. So now we can screen share, and then
This one.
this one.
Yeah. Or you can do the PowerPoint slideshow, either one. PowerPoint, because screen 2 is what's feeding out on the screen right now.
Yeah, we can just use that, right?
Okay.
So we will want that to be visible.
Yes.
Okay.
Levi, you want that to stream, correct? You want this to stream?
Yeah.
Okay.
When Jen gets here, she may give an introductory talk, so we'll have to switch out the presentation. But
It's okay.
Ravon, do you mind being on?
Sure.
We'll sit up here in the corner, like run up.
What is the mic? Can I teach you to do?
So now, do
Anything sweet.
you want to send that link to me so that I can just check if that's working, and then
That's a good idea.
let me just message that to me.
If you tap on it, maybe.
It's gonna.
this is surprise. Very big surprise. We made amends. Yeah. We have.
So it looks like, is it broadcasting now?
I had a friend tell us.
Yeah. Skip right here.
Ah, there we are.
Yeah.
Good. All right. Perfect. So then we'll just leave it.
And I think if we want to switch it to a different presentation.
He may, like when Jen comes to do the introduction, she may stand here too. Then
We'll just figure
so
it
that then the
Then-
camera is facing, or so that the view is on the person talking.
Oh yeah, so we can just say this.
Okay.
Okay, so this starts
Stop and start
yeah.
screen share.
Okay. And so then only if these are green means that they're not on. So okay, perfect.
Yeah,
so
right now
so
there's no
everything,
one there, but yeah.
So everything's on and it's picking up
Yeah,
our
this
sound
is what's broadcasting. So we'll switch to the presentation.
until he's ready.
Yeah, and then we'll wait on people come.
Okay. I think we're ready. Kristen's here. So then when Jen comes, you just switch it over here to the presentation, and then we can it's streaming now.
I don't know what you just said. It doesn't make sense to me.
We'll do it. Just
When Jen comes.
we'll fix it.
Okay.
Wait, don't touch anything. It's broadcasting.
Okay.
It's picking up your sound.
Okay.
It'll,
And have people tried to listen in?
yes,
But they can't. There's no feedback.
correct. We set it up just for.
So they're listening in right now. So watch what I say.
Yes.
Thanks.
Yes.
Thanks for coming. I would like to run through my slides. I'd like to run through my slides privately. That's not possible now. I'll just have to. Okay, correct.
And it's not safe every time.
And actually, so we go down, Darla, yeah, Donna. Yeah.
Usually set up the computer. I need to
Yeah.
see if
Plug in right here. She's gonna upload her.
Where's Gurban? I'm not gonna. How are you? Hey, Gurban. Gurban.
Yeah.
We're trying to figure out how to live stream, and I just got it right.
okay.
So I'll let the tech person handle.
Oh, cool. You're gonna do it from here.
Yeah, so it's broadcasting now. We have it set up so, but he will be your guide to.
Hi.
Do you want to show this one up on the?
I'm gonna do an introduction first.
Okay.
So
This so we're on Hangouts. So this is basically broadcasting live to a link somewhere. So you can show this there, and then you can choose between you talking on the camera, or you can have the presentation show up on the live stream. So
okay.
yeah.
I don't whatever you
Okay.
if we want to have the introduction live streaming, I'm happy to have that too.
Okay, cool.
But I don't know how to do it. So.
It'll always record your voice and it'll show up there. So
Okay.
even if you have slides, that's fine.
Okay.
So I'll just we'll probably show this up there, and then we can have.
If you want to just set it up, I'm just going to click through the slides.
Cool. So we'll just present this guy.
Ideally, if you can put it in presenter mode so I can see what's coming next, would be awesome, but
Got it.
doesn't have to be.
I haven't used this, so slideshow. Beginning. Oh yeah, it's showing up. Cool. So well, you can
Okay.
actually switch from here. So it'll work. Can you click on next, just to make sure it's going up?
I don't want to give away.
Oh no, it's actually not.
Let's see.
It might be a presenter.
There it is.
Cool. Awesome. I'm gonna do this little thing.
I'm gonna run to the bathroom.
All right.
As long as it clicks through, that's all I need.
Cool.
No, wait. Do you have? Is this, I don't know if it drifted or. So I have. Because I didn't see an HDMI.
I don't. There isn't an HDMI.
No, so I had to use a converter.
Do you want to try DVI?
What's DVI?
This guy here. What do you have for hookups?
All I have is HDMI.
Oh,
Yeah, so
I
that's
think
why.
I have a DVI to HDMI converter. The only problem is it's too fat to get this guy in there then too.
Oh no, it's okay. It's fine.
All right. Well, I mean,
It'll be
I
okay.
can try.
I'll just-
I just don't have a ladder or anything to.
It's not a problem.
Okay.
I think you. I think so. I don't know the login, but no, that was not.
Hello? Can everybody hear me? Great. Okay, well, thank you all for coming. Welcome to the defense of Levi Lewis's PhD. It's my pleasure to introduce Levi today. I'm his PhD advisor, Dr. Jennifer Smith. And as all of you probably know, Levi is a San Diego native. Uh-oh, hopefully this is gonna work. It was working before. Okay, I might need the IT guys back. Before we were just, oh, there it is. Okay, cool. So, Levi is a San Diego native, grew up here in San Diego County with his loving family, brother, sister, and mom and dad. It's clear from some of the photographs that I got of Levi when he was a child that he grew up in a very loving and nurturing family that actually, from an early age, knew that he wanted a career in academia. Levi's already planning to graduate at this young age, and he also was fortunate through the love and support of his family to spend a lot of time growing up on the beach. It seemed that he might have wanted to pursue a career in marine engineering initially, from this picture with him and his shovel, but then later he spent more time getting his hands and feet wet, playing with seaweed and other marine organisms, and it became clear that he was going to become a biologist. It became also very clear that Levi had a passion for fish. He loved fishing. He grew up fishing, seemed to take pretty much any opportunity he could to go fishing, as can be seen by numerous photographs that I found of Levi in the ocean, in freshwater, wherever he could, or kayaking as he so loves to do. Kayaking and fishing simultaneously. So it became clear that Levi really had a love for the ocean, a passion for marine organisms. But luckily, and he loved so much that at one point he thought he might even try to become a fish. I don't think the deep sea anglerfish quite suited him. So he went back to the sand and discovered his true passion, and this is a passion for marine invertebrates. So it became clear he had a curiosity of marine invertebrates at an early age, and this curiosity continued on into Levi's career, where he has spent the last greater than a decade focusing on understanding the natural history and ecology of these underappreciated and highly diverse marine invertebrates, some of which are called cryptofauna. So Levi went on, and his academic career began at UC Davis, where he did his bachelor's degree, spent a lot of time working in marine environments and estuaries, looking at the effects of excluding different types of organisms on marine community ecology. He then went on to San Diego State, where he did his master's degree, and apparently was quite passionate about his school there, and showed with the school mascot. So Levi then went on after graduating with his master's and joined our lab here at Scripps, or my lab here at Scripps. Although at the time, the lab looked more like this. So Levi was one of the founding members of the Smith lab here at Scripps. And through his adventures at Scripps, he started off swimmingly, where he received several accolades. He received several fellowships as he began his career at Scripps, receiving a Ford Foundation Fellowship. He was an Eigert Scholar. He was a San Diego Fellowship recipient, and received numerous awards for conference attendance and for excellence in his research. Part of his research experience here at Scripps began with his adventures off to the Hawaiian Islands, where he spent much of his time on the island of Maui conducting the majority of his PhD research there. And he embraced the islands and learned a lot, spent a lot of time working around the Hawaiian Islands, but Maui in particular. He was a founding member of what we call SLIMEO, the Smith Lab Interim Maui Extension Office, as you can see them starting the SLIMEO lab there. He then continued and became a master of conducting research on Maui's coral reefs, whether it was using photoquadrats, carrying his dive buddy's dive gear as it's falling off of her, conducting lab experiments, and became a master of just experimental design, designing all kinds of crazy, crazy experiments, which Levi definitely became a master of, as you can see here. Pretty much any way, shape, or form that you could put a cage underwater and exclude or include things, he pretty much figured out. Some of these seem to have taken inspiration from Star Wars creatures, but Levi pretty much is very creative when it comes to designing experiments underwater, and spent a lot of time doing that. Along his way, he really became a mentor for the lab. Levi brought more students, undergrads, master's students, starting students into the field with him and taught them how to conduct scientific research underwater. And this was much appreciated from me, but from the whole lab as a whole, really, because he was a very important mentor for all of these students. He also embraced Hawaiian culture, and with that means embracing food and drink. As you can see, Levi is always taking advantage and enjoying any opportunity he can to eat shave ice or have a BFRD, or more importantly, probably I don't know if this is where the love for Spam first originated, but it definitely took hold, where Levi became a very serious Spam connoisseur, especially with his friend Clint, who you can see here holding an entire can of Spam and a fork. But this was most enjoyable when I get an email from them saying that they had discovered Spamadamia nuts, which were Spam flavored macadamia nuts, and they were very excited by this find. So they continued their love for Spam, and it seemed to be a nutritious source of energy for them in the field. Levi also mastered opportunities to fall asleep, and it seems that Levi could fall asleep just about anywhere, anytime, any place, given a minute of downtime. So while food is important, it is also important to catch a few Z's while you're out there doing all of your field work. So Levi mastered that as well. He also enjoyed taking part in a variety of beach-affiliated games such as bocce or cube, as we will hear more about later this afternoon, hopefully. Even if bocce took place on the top of a volcano, as seen here on the top of Haleakala. Levi made friends with many different organisms, including sea turtles. He also made friends with a lot of the local community. This is a large group of people on the island of Maui celebrating the annual birthday of the establishment of this really important marine protected area. And the lab always takes part in these celebrations, an important way to integrate and celebrate this achievement with the local community. And this also always involved the baking of various items that celebrated this marine protected area. And Levi was the inventor of the phase shift cake, which is showing a transition from a degraded reef back to a healthy reef. And each one of these cakes is a different year. So you can see that Levi was very dedicated to his pursuit in the perfection of the phase shift cake, which he received awards for even because of the creativity. So we spent a lot of time on Maui conducting research, celebrating, having a good time, and just generally fostering a good environment. Levi's been an integral member of the lab family and has conducted a lot of great work along the way. He also was fortunate enough to be able to attend or be a participant in a Native Hawaiian and scientific and cultural cruise to the Northwest Hawaiian Island. Levi took place in this cruise where they surveyed the intertidal, often using climbing gear on these really extreme habitats. And he participated in those cruises for at least three years. He also was fortunate enough to go to the Maldives, where he conducted research at a new marine lab there, giving scientific presentations to the local university. He's participated in numerous conferences around the world, including the International Coral Reef Conference, both in Cairns, Australia, as well as in the Hawaiian Islands just this last summer. So Levi has also earned an honorary PhD in how to travel the world while conducting his scientific research. He's made it around pretty much all of the oceans aside from maybe the Caribbean, doing research or giving talks at conferences. And he's really excelled along the way. Levi has published 6 peer-reviewed publications already. He's received numerous awards for his work. He already has a postdoc lined up at UC Davis, and we're really excited to see him move off to his next adventure. He's been a really important teacher here at Scripps, teaching both phycology for me as the TA, and teaching many local school groups, whether it be at UCSD or local communities, other small schools, high schools, community groups. He's also been an important member of many different types of athletic groups, whether it be when he was at Davis, I think this is dodgeball, softball, volleyball. You can see he takes his sport seriously. During his PhD, Levi also perhaps caught the best catch he has yet to catch, and that's not this fish, but that's his fiance. So Levi met Tiff along the way and is now engaged, and we look forward to their future together. Again, he's been a really important member of the lab family, had a lot of great times along the way, hopefully learned a lot. You'll hear a lot more about his adventures. He might not always be happy, but nonetheless, he always seems to embrace a challenge, whether it be on land, in the sea, or even on top of the world. Levi has been a great part of the lab, and I look forward to seeing where he goes in the future. And with that, I would like Levi to tell us about his PhD work. So thank you. That was easy.
Let's see. I gotta figure out how to get out of here.
Apparently we needed.
We're live streaming, so let's see.
It's all good. Oh, you
It's not advancing.
Oh, got it. So
Yeah,
you
yeah.
would have to switch to the actual presentation, which is right here. Yeah, and then
Okay.
give me a sec.
We're live streaming for a lot of the Maui family that can't be here. So there are many people tuning in, and there are other researchers all across the country that I've worked with that are also streaming in live right now as well. So thank you, Baton and Pravon, for setting us up. I appreciate it. And Jen, thank you so much for the introduction. That was amazing. So before I continue on, I'd like to acknowledge my chair, Jennifer Smith, and the rest of my doctoral committee. So Drs. Andres Anderson, Jim Lecter, Lisa Levin, John Shuren, and Stuart Sandin are all here, and who've all provided excellent guidance and critique of this body of work, and I'm looking forward to getting even more afterward. So I'm getting my degree in biological oceanography, but really I'm going to be talking mostly about marine ecology, and specifically coral reef ecology. And ecology is the scientific study of the structure and function of nature. And it's also the study of the relationships between organisms and their environments. And there are a variety of different. Sorry, we're having some more technical difficulties with the streaming. Ron, could you take one more look at this? We might have to go off the live stream. Yeah,
So I can see you on the screen.
it's advancing here, but it's not advancing.
Oh, got it. So
Yeah.
I think.
Sorry about this.
Let's see. Yep. I think we're back in action. So this is the study of the structure and function of nature, or the relationships between organisms and their environments. That's you. And there are several classic constituent theories within the overall theory of ecology. One is succession. The idea that bare space is occupied first by organisms that can disperse and reproduce very quickly, and therefore colonize that space first. However, those organisms are typically not equipped to compete with other organisms. So through time, other organisms will eventually colonize that space and outcompete the existing organisms as early dispersers. And that through time, you'll see systems will change from, for example, early colonizing annual plants, going from left to right, to shrubs, to trees, and to climax communities. And the only way in which these early colonizers persist is when there are disturbances that interfere with the climax communities.
And in fact, when there are intermediate levels of disturbance, we get higher levels of diversity. And the reason is we have both climax communities and these early colonizing communities and everything in between all coexisting together. And the concept of succession really basically suggests that two species that are competing for the same food source or the same resource, which could be space, will compete with each other, and one of them will be a superior competitor. And it will drive the other one to extinction. And so again, if we have disturbance, we see that the dominant competitor can no longer drive the other to extinction. But another way in which species can coexist is by diversifying their niches. And their niche is the suite of ecological resources that they use. And so, for example, Charles Darwin talked about finches in the Galapagos and pointed out that they develop different sized beaks to exploit different sized prey. So we have multiple species all coexisting and not necessarily driving each other to extinction. And the reason I bring these up is that I study coral reefs, and many of these processes are also occurring in coral reef ecosystems. We have succession that's leading to different ecological states. One state could be coral dominated, another one could be fleshy, algal dominated, and these two different organisms are sessile organisms that are competing for space on the reef. And there are different factors that are driving the succession into different ecological trajectories. So disturbances such as grazing by herbivores or sedimentation or even the input of nutrients can all change the successional trajectories. Of course, there are also larger acute events that can occur that are not deterministic but are more stochastic that can also disrupt some of these processes. And furthermore, coral reefs are some of the most diverse habitats on the planet. They contain 1/3 of the species in the ocean, though they only contain around 1%, or they only cover about 1% of the benthos. And so there's a great opportunity for the diversity of species in these habitats to have developed different ecological niches. We know that top-down effects, or the effects of consumers in many different ecosystems, are really important. And early work by Payne and colleagues looking at the effects of sea stars in the intertidal zone really demonstrated this very strongly. And so we have carnivores eating herbivores and herbivores eating producers, and the interactions between these kinds of dynamics in food webs and food chains. When they're strong, we call them trophic cascades. And it can look like a lion and a gazelle and a grass, but it can also look like a fish and an invertebrate and a kelp, or it can look like a sea otter, an urchin, and a kelp in the ocean. And this is one of the most famous trophic cascades documented by Estes and Palmisano. Based on a suite of published literature, we know that ecosystems, certainly the marine benthic ecosystems, seem to have some of the strongest top-down effects on the planet. Randall Hughes looked at the literature on seagrass ecosystems and demonstrated that experiments showed that grazers had this strong negative effect on algal biomass, whereas the increase of nutrients really didn't have much of an effect. So indicating that consumers, these things that are eating the algae, have the strongest effects, more so than these bottom-up effects such as nutrients. And one of my committee members, John Shuren, published work examining the strength of trophic cascades across many different ecosystems, looking at lakes and rivers, terrestrial environments, and even marine pelagic ecosystems, and concluded that the top-down effects in marine benthic ecosystems are some of the strongest. Paul Dayton, working in Antarctic, described a community of organisms that he called foundation species. And we tend to think of one aspect of his definition, specifically with respect to these sessile foundation species, organisms that we call ecosystem engineers, organisms that settle and grow, they're sessile, and they create a huge amount of structure or complexity in an ecosystem that otherwise would be fairly simple. So he first was talking about these sponge communities, but we can see similar types of important ecosystem engineers in the terrestrial realm, such as trees in the forest, kelps in a kelp forest, and coral reefs on a coral reef. And I've worked both in seagrass beds and coral reefs, and these are two very different systems. Seagrass beds are plant-dominated with high nutrient, soft bottoms, and small grazers. And coral reefs are pretty much the opposite. They're animal-dominated, low nutrients, hard bottom, with large grazers in general. But what's interesting is that the dynamics in these two systems are fairly similar. We have grazers consuming early colonizing algae that are competing with the foundation species. And the same for coral reefs. We have grazers that are consuming these early colonizing fleshy algae that are competing with the foundation species. And in both of these systems, these grazers are thought to have positive indirect effects on the foundation species. And importantly, the effects of humans potentially on these trophic dynamics in these systems are actually different. So for example, humans have much more diffuse effects that have to go through multiple levels in seagrass ecosystems, but have much more direct effects in coral reefs. And so I've published work on top-down dynamics in seagrass beds and looking at different ecological functions of grazers in seagrass beds. I've also published work with Jill Harris looking at these early successional colonizers, these filamentous algae in the Maldives, and with Jen examining how the presence of humans might impact the successional state on a given reef system all across the Central Pacific. And so the general theme of my academic career is thinking about top-down effects of consumers on these ecological processes. And so for my dissertation, I've chopped it up into two themes. Theme one, I'll be looking at spatial variation in herbivore communities and their effects on Maui, Hawaii. And the second theme, I'll be looking at differences in the ecological functions of the dominant herbivores in these shallow reef systems on this island. And in Hawaii, in the shallows, the dominant herbivores, as I'll show you, are these echinoids. And real quick, the words we use reflect the way in which we see the world. Echinoids are also commonly called urchins, and Jeremy Jackson pointed out that urchin used to be a derogatory word for a poor street child, not unlike calling them little rascals. But the original word came from a French word for hedgehog. And so when you think about it, echinoids or sea urchins, and I'll probably use those interchangeably naturally, could also be considered sea hedgehogs or even sea rascals. So I just gave you a quick introduction to this thesis. Next, I'll talk about theme two, looking at comparative experiments and spatial variation around Maui. And last, I'll look at echinoid metabolic diversity, trophic diversity, and functional diversity to get a better understanding of the functional diversity within this really important herbivore guild. And these chapters all contribute or are founded upon various constituent theories within the general theory of ecology. So hopefully they'll contribute and inform us a little bit more about foraging theory and succession theory and populations and niche theory, niche theory, and food webs, and also metabolic theory, all relating to these really important echinoid grazers. So theme one starts and ends with chapter two. So I'll be looking at spatial variation in herbivore communities and their effects in Maui, Hawaii. So the reefs in Maui, Hawaii are really important. They're highly valuable. In fact, recently they were valued at $38 billion to the American people. They're important for a variety of reasons. And I've promised people that I'd provide short snippets of poetry. And so I have a quick piece of poetry to describe why they're important. So coral reefs are important because the corals build reefs, give homes of relief from massive waves and surgy seas. They cure disease, make food to eat, and fuel the nearby fisheries, maximize complexity, highest diversity in the sea, and most importantly, all their services are free. So as I described before, we have organisms living on the seafloor, and they're competing, and we have producers and these foundation species. The corals are the foundation species, creating calcium carbonate rock, which builds the entire reef structure, and they're constantly competing with other organisms, such as algae, that are also trying to occupy space. And these dynamics are being affected by top-down effects. We have some predators, like this triggerfish here, which, by the way, is the humu humu nuku nuku wapa'a, with a degree studying Hawaiian species without being able to say that name. But there are actually a lot more grazers in this system. We have fishes that are grazers and also these herbivorous echinoids that are grazers, all of which are consuming algae. And generally, these grazers are important because, again, they consume algae, which therefore ultimately result in positive and direct effects for the corals. And the corals are the foundation species. In contrast, we have bottom-up effects, such as the inputs of nutrients that fuel algal growth, which actually can result in negative and direct effects on the corals. And we have abiotic effects, such as inputs of sediment, that can directly harm the corals. And so all of these dynamics are occurring in the Hawaiian Islands all the time. And more importantly, humans have strong effects on each one of these dynamics. So for example, we alter the upland terrain through agriculture and development, which increases the sedimentation into the system, which harms the corals. We harvest the fishes and the urchins for food or for the aquarium trade, which ultimately leads to negative effects. And we also inject lots of wastewater into the nearshore environment, which fuels algal growth also causing negative effects on the corals. And so it's not surprising that around the Hawaiian Islands, in particular around Maui, we see rapid declines in coral cover at many of these sites circled in red. And in fact, we see this pretty much around the world, where coral cover, these small green dots are sites all around the Indo-Pacific and the Central Pacific, and we see a general trend of coral cover declining. And islands with people actually are doing worse than islands without people. And several studies from our lab have shown that. Stuart Sandin showed that islands with the highest coral cover and the top predators generally do not have people. So these sites are like Kingman Reef and Palmyra Reef in the Central Pacific. And Jennifer just recently published a paper showing that though there's a lot of variability in what kinds of reefs you might find, both populated and unpopulated, all of the reefs with the highest calcifier cover, including corals and other calcifiers, are almost entirely all unpopulated. So the presence of people seems to have a really negative effect on coral systems. So what I wanted to do for this chapter was to think more about what we know and what we might be able to find out. We currently know a lot from extensive monitoring all around the Hawaiian Islands about trends and correlations and health indices with respect to coral cover. However, we often don't test or measure the actual processes that are potentially driving those patterns. And so I chose to do a manipulative experiment that could help us understand how not only the patterns vary, but also potentially how the processes vary. And the goal was to better inform future management decisions for the reef system. So the overarching questions that I wanted to ask was, how does reef development vary spatially along Leeward Maui? How do consumers and their effects vary spatially? And how does environmental context relate to reef development and the influence of consumers? So we conducted an experiment along Leeward Maui, and we selected 8 sites that spanned the entire leeward side of the island. And at each of these sites, we conducted an experiment by outplanting bare surfaces that would serve as surfaces for colonization so that we could examine succession. And we did some preliminary surveys, and we saw significant differences in the communities at each of these different sites, going from north to south, 1 through 8. And so we can see that coral cover in the red varied, turf algae in the light green varied across sites, and we see that macroalgal biomass also varied. And each of those are important functional groups that we'll revisit multiple times. The question is, why might some of those different patterns exist? So our approach was to characterize each of these 8 sites according to herbivores, nutrients, and sediments, the 3 different factors that I've already introduced, and then to compare succession on these tiles with and without exposure to consumers. So we have some tiles that are open to consumers and other tiles that have a cage, which was designed by Don McLeish in Maui. And at each of these 8 sites, we had 10 uncaged or open tiles and 10 caged tiles. We repeated this experiment across all 8 sites, and then we examined development on these tiles between sites, between treatments, caged and uncaged, and in relation to the environmental metrics that we measured here in 1. And again, the goal of the comparative experimental method is to repeat an experiment at multiple locations to improve our understanding of the general applicability of the results that we find. For example, if we did an experiment at one site, which most experiments occurs as one site, we might come up with, or we might find or make conclusions that aren't actually relevant at other sites if we had repeated that experiment. So first, I'm going to talk about how these different sites varied in terms of their environmental characteristics, herbivores, nutrients, and sediments. So these are the data from herbivore surveys. We did 4 surveys at each of the different 8 sites, and the error bars aren't on there, but they're relatively small. We saw a large variation among sites in total herbivore biomass, ranging anywhere from around 20 to 600 grams per meter squared, and these values are similar values that have been observed in other reefs, for example, in the Indian Ocean, in particular shallow coral reefs. And most importantly, we have both echinoids and fishes stacked here, and what you see is that fish biomass is actually relatively small. So these systems really truly are dominated by echinoids, and most studies of herbivore dynamics, or many studies of herbivore dynamics, really focus on the fishes, where in our system we really need to focus on these spiny echinoids. We also examine nutrients in terms of dissolved inorganic nitrogen, so different species of nitrogen. And these are the values. We have nitrate, nitrite, and ammonia combined, and we see the values range from around 0.4 to 1. And those are all actually in relatively normal or healthy levels for coral reefs. And we also examine sediment accumulation rates at each of these different sites. So this is the exposure of of the sites to this level of abiotic stress. And we see very large variation in sediment exposure. And these are just some baselines for reference. So for example, according to Clint Edwards' paper in our lab, pristine fish biomass is around 40 grams per meter squared. So these echinoids are actually really abundant, and there's very high biomass of echinoids at each of these sites. In contrast, nutrient stress thresholds, according to various researchers in the Caribbean, are suggested to be around 1 micromolar. And so most of the nutrient values are below that, indicating that these systems aren't necessarily in a nutrient stress state. And in contrast to this, the sediment stress threshold proposed by Rogers in the 90s suggests that 10 milligrams or 0.01 grams of sediment accumulation per centimeter squared per day is stressful for reefs. And the majority of our sites surpassed that threshold, most of them by several orders of magnitude. So these systems were heavily sediment stressed and echinoid dominated. So next, I'm going to talk about what occurred on the tiles. So we'll start off by talking about macroalgae. These are the results of macroalgal biomass. So we collected the tiles from the field after 3 years, and we looked at the total amount of macroalgae that had accumulated on the tiles. We have both the open tiles and the cage tiles that were excluded, where consumers were excluded. We have the sites 1 to 8, and we have macroalgal biomass in grams per tile. And the first thing you notice is that there's large variation among sites, even when they have consumer access in the gray bars. So these are just 2 examples at Puamana and Maalaea that have different values for macroalgal biomass. We also see large variation when consumers are excluded. And what this suggests is that there's natural differences in production of algae. This is not a consumer effect. This is just naturally different algal production in the system. And furthermore, we observe significant differences in the effect of consumers on macroalgal biomass. For example, here at Uluwatu and also at Maalaea, we see very small differences between gray and black, or the open versus closed cages. In contrast, at Kavaku, we see a very large difference between the two, indicating a strong effect of consumers. So sites differed markedly in algal productivity and also in the effects of herbivores on total algal biomass. Furthermore, we can explore how macroalgal biomass on the tiles correlates with several of these different environmental stressors or environmental factors. So for example, herbivore biomass, nutrients, and sediments. And we might predict that as herbivore biomass goes up, we'd see macroalgal biomass going down, especially on the open tiles. We might expect nutrients to increase macroalgal biomass, especially in the cage tiles. And sediments overall, just one possible outcome could be a smothering effect of sediments on macroalgae. And these are the results, however, and we don't see any effects of any of these environmental characteristics on macroalgal biomass. And so that's what we concluded, is that it doesn't appear, given this experiment, that macroalgal biomass was strongly driven by any one of these in isolation. But we can also look at the effects of herbivores, remember. So that's the difference between the gray and the black bars. And so we can create an effect size using a log response ratio, where values above 0 indicate that consumers increased algae, which would be odd, and values below 0 indicate that consumers decreased algal biomass, which is what we would expect, and at the red line, 0. And these are what the data look like. So most sites observed a reduction in macroalgal biomass. However, some sites much more so than others. And overall, there was around a 3-fold increase in macroalgae inside of these exclosures. We examined herbivore biomass and its effect or correlation with macroalgal biomass, and we saw no significant effect. However, when we look at the effect size or the effect of consumers across these different sites by comparing cage versus open, we actually do see a significant effect and a relatively strong significant effect of herbivores. So what this suggests is that doing simple observational studies might miss some of the important dynamics that are occurring in the system. For example, macroalgal biomass isn't only driven by consumers, it's also driven by other factors that might determine its productivity. So next, we wanted to look at not only macroalgal biomass in bulk, but also examine what does the community look like on the surface of the tile. And this represents the surface of the reef community as well. And so to do so, we used 50 stratified random points, and under each point, we identified what organism was dominating that region of the tile, and then we estimated a percent cover of the key functional groups on the tiles. And we conducted a pretty complex multivariate analysis examining variation in the communities. I'm not going to go into it, but there are some really interesting patterns that I would love to discuss further. But for this talk, I'm going to keep it a little bit more simple. And again, we have these sites going from 1 to 8, north to south, and now we have percent cover. So the percent cover on the tile of these different functional groups. And to simplify even further, we're only going to focus on 3 very important functional groups for reef systems. 1, fleshy macroalgae. 2, crustose coralline algae. And 3, the turf algae. So macroalgae are large fleshy algae over 1 centimeter in height that strongly compete with corals. Crustose coralline algae are crustose, so they're flat, calcified algae that oftentimes actually serve as important settlement substrates for corals, so that you could think of them sometimes as actually good for corals and certainly good for calcification on the reef. And then turf algae are thin filamentous algae that also compete with corals on the reef system. And so what we're going to do is just look at consumer effects. So again, we're looking at the difference between open and cage tiles and inferring whether or not consumers had positive effects, no effect, or negative effects on these different functional groups on the surface tiles. And so these values here are going to be the change in percent cover between open, or the difference in percent cover between open and closed tiles. And first, we're looking at macroalgae. And overall, we see a general decline in macroalgal cover when consumers have access, and that's what we would expect. However, there is a lot of variation among sites in those data. With respect to CCA, we don't see that same pattern. We see, again, variation among sites, and overall, no overall effect across all sites on these crustose coralline algae. And last, we see increased turf cover as a function of herbivory. So herbivores actually increase the abundance of these filamentous turfs throughout most of our system, in case it was the strongest effect that we observed. And this is interesting for several different reasons. Primarily, the classic relative dominance model suggests that herbivores should actually increase the amount of crustose coralline algae because these algae are resistant to herbivory. And in contrast, these consumers would reduce the amount of turf algae. So we're seeing the opposite pattern of what we might predict based on a classic model. And that's something interesting that I'll touch on in a minute. The last metric we looked at was coral recruitment. So how many corals are we observing in the system on each of the tiles? And these are the data for those. We have coral counts, coral recruits counts on each cow. And the values range anywhere from 0 up to around 8 individuals per tile. And we saw significant differences among sites, but not significant differences as a function of caging. So cages didn't affect the abundance of these coral recruits. Perhaps most interesting was when we looked at sediment accumulation rates, or sediment exposure. Strong negative exponential relationship between the number of coral recruits, now converted into recruits per meter squared at each site, and sediment accumulation rates. So what this suggests is that the overarching, or the strongest effect of any environmental characteristic was the degree in which they were being exposed to sediments. And also interesting, at one site that had very low sediment exposure, and only at that site did we see a potential effect of consumers. In this case, a positive effect where consumers increased the amount of coral recruits. So that, again, that was only at one site. So in summary, at these sites, herbivory was echinoid dominated. There was large variation among sites in the potential algal production, and consumer effects were relatively strong when we examined them experimentally. All sites were sediment stressed. Coral recruitment was highest with low sediment exposure. And again, that one site with low sediment exposure suggested that these urchins might actually have positive effects, but again, only at that one site. And there were several other sites that actually had low sediment exposure rates and still didn't have high coral recruitment, suggesting that there's something else at those sites possibly limiting coral recruitment. Furthermore, as I mentioned earlier, herbivores were unable to shift the site from a fleshy algal-dominated system to a crustose coralline algal-dominated system. And so this might suggest that a sediment stressed or sediment suppressed herbivory model, where sediments prevent herbivores from being able to create dynamics that would lead to these dominance of calcified coralline algae, might be more appropriate for the system than the relative dominance model would suggest. So that was the end of theme 1. And the rest of the talk will be thinking more about biodiversity and ecological function of these really important echinoids. So previous work in the terrestrial realm has demonstrated a really strong effect of biodiversity on a variety of different kinds of ecological functions. For example, using a variety of agricultural plots, David Tillman showed that as the number of species in a plot increased, the total amount of biomass also increased, or the biomass that was produced in the plot also increased, suggesting that there's a positive diversity effect reduction. And this has been observed across many different systems, and a paper was published fairly recently pointing out that it appears that these biodiversity effects may be stronger than a variety of other types of environmental considerations, such as resource availability or disturbance, or even herbivory. So this has been measured in many different systems on many different processes, and again, we often see very similar patterns in these really strong, pervasive effects of biodiversity. Diverse consumer communities appear to have stronger effects than less diverse consumer communities. For example, Doug Rascher showed that as herbivore diversity increases here on the x-axis at sites that were reserves or protected from fishing, we see a significant reduction in macroalgal cover, suggesting that these more diverse herbivore communities, in this case they were fishes, have a stronger effect on macroalgal cover. And an experimental study by Burkepile and Hay demonstrated that when 2 different species of fish were included in a cage, we saw significantly higher coral cover or coral growth due to their enhanced ability to control macroalgae. However, studies like this haven't really been done for these echinoid communities, as I've shown, are really abundant and dominant around these shallow reefs in Maui. And so one of the questions I addressed was, how do these different species function on the reef? And do we see similar positive effects of diversity? We know echinoids are important from other studies in other systems. For example, in the Caribbean, a massive die-off in the 80s of this urchin, and it was a die-off that was Caribbean-wide, resulted in the largest and quickest phase shift ever documented in a reef system. So this urchin died across sites spanning the entire Caribbean, and the importance of this urchin became wildly apparent when the system transformed from a coral-dominated system to a macroalgal-dominated system. And the importance of these echinoids are actually being used in different systems to try and increase coral cover. So for example, in Hawaii, sea urchins are being bred and outplanted on reefs to control these algae and to promote coral growth. And the same is being done in the Caribbean using diadema, the urchin in the 1980s. However, not all urchins are necessarily good for reefs. For example, in Kenya, in the Indian Ocean, this urchin, Echinometromathii, has actually been shown to harm coral and to eat calcified algae and actually have negative effects on reef accretion. And other species have been shown to do the same. In fact, they've shown that the presence of a predator actually has a benefit for the reef system because it controls this urchin. So what I asked was whether or not these dominant echinoids around Maui exhibit unique characteristics and unique functions. And so the next chapters are going to focus on metabolic diversity, trophic diversity, and functional diversity of these 5 species of urchins that are common in Maui. So for chapter 3, realizing that metabolic rates are really important in ecosystems, we first examined whether or not these different echinoids have different metabolic rates. And this is work that I conducted with Joanna Einad and of course my advisor. And the rate of metabolism sets the pace of life, as said by James Brown, but it was actually a different James Brown from New Mexico, who's one of the leading metabolic theoreticians in the country. And the metabolic theory of ecology, or MTE, has demonstrated that using very simple models, really just incorporating temperature and mass, we can explain a huge amount of the variation in production and metabolism across really large diversity of organisms, plants, mammals, birds, from plankton to whales. And so it's a really powerful tool to think about ecology. And Ryan Heckinger taught just last year a really nice seminar course where we went through some of the major tenets of this theory. And so that inspired me to think more thoroughly about how these echinoids might differ metabolically. So we conducted a series of metabolic assays at the Maui Ocean Center. So we collected urchins over here in Maui and transported them to the Maui Ocean Center. And then we conducted a series of metabolic assays on these urchins, examining their respiration rates, which then we inferred as their metabolic rate. And we did some allometric scaling. This is just the allometric scaling of mass, and we showed that these echinoids have differences in the way in which their mass is scaled, basically based on the differences in their spine morphology. And more importantly, we demonstrated that there were differences among species in their metabolic scaling, indicating that they have true differences in their metabolic rates. And these differences and the slope of these lines was significantly different from what classic metabolic theory suggests, which would be a slope of 0.75, and the slope that we found was 0.44. And so that suggests that classic standard metabolic scaling may not apply to these urchins, and for using metabolic theory, we need to think about how this group might differ from classic scaling relationships. And so fundamentally, we showed that different species have markedly different metabolic rates, and that these differences in metabolic rates might infer differences in their ecological function. And so that brings us to chapter 4, where we examine differences in their grazing behavior and how that relates to differences in their metabolism. So we conducted a series of grazing assays out in the field. We set up little enclosures and provided different bits of algae to urchins and just examined how quickly they consumed and what kinds of things they preferred to consume. And the overarching questions we asked was whether or not echinoids differ in their grazing rates, whether those grazing rates correspond with their metabolic rates, whether echinoids exhibit these unique preferences for different kinds of algae, and whether or not the preferences for different kinds of algae also correspond with differences in metabolism. So again, we looked at these 5 species of urchin, and we presented each of these with 6 different kinds of algae, each representing a really important functional group, from fleshy, red, blooming invasive algae all the way to calcified crusts and thin filamentous turfs. And our field-based assays are an important advancement in the field of trophic ecology, because most assays are conducted in the lab where organisms are either starved or have been conditioned by holding them in the lab and feeding them. And in this case, we've actually given the urchins no pre-treatment. They're taken directly from the reef, and we're looking at what their grazing behaviors are like in the field. So again, we use these little, I think Clint called them my urchin prisons. And we placed the 6 different kinds of algae in an array, added the urchin, and then allowed them to consume. And then we looked at how much of the different kinds of algae that they consumed and quantified the total amount of material that was removed. We also had controlled cages to account for any losses just due to the experimental design. So these are all going to be control corrected values. And so this first graph shows the total grazing rate across the 5 different species of echinoid, ranging anywhere from less than 1/2 gram per day to around 5 grams per day, with lots of significant variability. And on the left, we have the total or individual grazing rate, and then we also examine the mass-specific rate, correcting for differences in size among the different species. And in both cases, we see very large and very significant differences in grazing. When we compare those to independently measured metabolic rates on different individuals, we see almost the exact same pattern, both for total individual metabolic rate and for mass-specific metabolic rate. And so when you plot those together, we see that total metabolic rate, again measured on different individuals, at the species level explains 98% of the grazing rate, indicating that the grazing rates we measured are strongly and physiologically regulated and differ strongly among species. And a similar pattern was observed for the mass-specific rates. So again, species differences in grazing are physiologically driven and taxon-specific. So next, we're going to look at differences in algal consumption, preference, and variability in preference. So we examined not only the total amount of material that was consumed, but also these are the urchins from left to right, but also the different kinds of things that they prefer to consume. So this is a measure of percent consumption, how much of each type of alga the echinoids consumed. And I failed to mention, these are averages of 20 individuals. So we did 20 individuals of for each of these 5 different species. And this is a little bit messy to look at. So what we can do is take the maximal consumption rate, or the consumption rate of the maximal or most preferred organism. For example, in this case, it's the blue Acanthophora, and we can call that a preference of 1. It's the most preferred. In contrast, for Trypnustes, TG, brown, Turbinaria is the most preferred, and we call that a 1. And we can scale all other algal grazing rates to the most preferred consumed alga and provide what I call my grazing preference heat map. And this is just a hypothetical heat map showing what we might expect. So for example, if all individuals ate all algae equally, we'd say that they all are a 1. They prefer every alga 100% the same. And that would be non-selective uniform grazing. But also every individual of the 20 individuals might eat a different alga, in which case you would also say that's non-selective grazing, but there's lots of individual variability. And that would look like this. It'd be a lower preference score across all the different kinds of algae. And then a moderately selective would be a patchwork, and a highly selective urchin would only prefer one type of algae across all individuals, and it'd be very strong. So this is what we see. We see for Trypnustes, it's the only urchin that prefers to eat Turbinaria. Turbinaria is a very robust and chemically defended alga that none of the other urchins prefer to eat. And this actually corresponds with some aquaculture literature indicating that this species, for whatever reason, loves to eat these chemically defended algae. For Echinopteryx diadema and Echinopteryx calamaris, these are 2 congeners. They showed very similar patterns. They prefer to eat the invasive blooming Acanthophora and also the blooming ulva. And they ate some of the others, but really preferred these 2 individuals. For HM and EM, Heterocyntrotus and Echinometra, we kind of see that gray pattern indicating no strong selection and a lot of variability among individuals. Well, we can't see what the individuals look like in this kind of average plot, but we can actually plot what all the different preferences of individuals look like using some standard ordination techniques like non-metric multidimensional scaling. And we conducted a permutation analysis that indicated there were significant differences in these preference profiles of these different species. So you see Echinopteryx calamaris and diadema in the blue triangles and the blue circles clustered very tightly and closely together and were significantly different than Trypnustes, which is in the purple here. And Trypnustes was feeding again on these Turbinaria. And perhaps even more interesting is the size of the circles, which are indicative of dispersion and variability among individuals. And so these have very low dispersion, and Heterocyntrotus, HM, in the red triangles, Metra in the yellow triangles, had very large circles indicating very large dispersion and very large variability in terms of preferences. And in fact, when we plot preference dispersion, or a measure of the size of the circle in relation to metabolism, we get an interesting pattern. We see significant differences in preference dispersion, and those seem to correlate with metabolic rate, such that low metabolic species have high dispersion, which would indicate low selectivity, and individuals, or the 3 species with high metabolic rates, exhibited low dispersion, indicating higher levels of selectivity. And this matches optimal foraging theory, suggesting that these species with higher metabolic rates need to eat higher quality foods. And also, they've developed methods, or their activity allows them to pursue and to search and travel to find these higher nutritious foods. So in summary, we observed large differences in grazing and metabolic rates. Metabolism explained 99% or 98% of the difference in grazing rates. Lower metabolism appears to correlate with lower selectivity and higher metabolism of these larger, more robust fleshy algae. And we observed, even within these high metabolic species, we observed significant differences in the types of algae they prefer to consume. So for example, difference where TG preferred Turbinaria and Echinopthrix preferred Ulva and Acanthoptera. And so last, we also not only wanted to know, do these urchins differ in their own sake, but do these differences confer different consequences on the reef itself? And so do we see different species exerting different effects on these dynamics, macroalgal consumption and coral growth? And do we see diversity, so increases in the numbers of species, also affecting these dynamics? This brings us to chapter 5, where we examined community composition and how it affects benthic dynamics on a natural coral reef. And this is work that I had conducted with Clinton Edwards, and also my advisor. So we're still working in West Maui, and we established an experiment consisting of a randomized block design. We had 8 different treatments, 5 different spatial blocks, and 40 individual plots. They were on 1 meter square reef isolates at around 8 meters or 20 feet, 20 to 30 feet of depth. The experiment ran for 24 months, so 2 years. And these are what the overall treatments look like. We had open unmanipulated plots where we had natural abundances of herbivores, both fishes and echinoids. We had full exclosures where no herbivores were allowed in. And then we had 2, these were procedural controls. And then we had 2 different diversity treatments, one that was monospecific, so 2 individuals of the same species, and one that was multi-species, 2 individuals, each from a different species of echinoid. And so the overall design looked like this, where we had our 2 controls, full exclosures and open plots with ambient levels of consumers. Our 3 monospecific treatments, where we had Heterocyntrotus, Echinopthrix, and Trypnustes, and single species treatments, and then all pairwise combinations of these 3 species, TG and HM, EC and HM, and EC and TG, in our multi-species treatments. And these are what those look like. So this is a picture of the cage. This is a multi-species treatment with 2 species, 2 of the same species, and a single species treatment, and again, full exposure. The overarching questions that we wanted to ask are whether or not echinoids differ in terms of their ultimate effects on benthic communities, in particular algae and coral. Do more diverse communities, in particular 2 species in this case, exhibit stronger effects than single species communities? And do specific community types exhibit stronger effects than other community types? The metrics that we measured are as follows. We looked at the density of macroalgae and the heights of turf algae, those thin filaments. We also examined the total biomass of accumulated macroalgae at the end of the experiment after 2 years. And perhaps most importantly, we examined changes, or the delta between initial and final, and the aerial cover of dominant groups, again, in particular macroalgae and corals. We'll start off looking at macroalgal density and turf height just briefly. So these are examples of macroalgae. We have Laurentia, we have Asparagopsis. These are more of the larger, robust macroalgae that grow typically only when consumers are excluded. And these are some of those filamentous turfs that we described previously. So what I want to point out in these 2 figures, and I'll walk you through them in just 1 minute, is that there were no initial differences. So we have initial in this column, after year 1, after year 2, and that there were large differences among these different treatments, and the treatments go from left to right for each of these years at year 1 and year 2. So you can see the size of these different polygons show the amount of variability among treatments, and we see no differences here, large differences at year 1 and year 2, no differences in turf height, large differences at year 1 and year 2. We collected macroalgal biomass as well, and I'm not going to go through the details of these data because they look very similar to macroalgal cover, not surprisingly. But the most important thing I wanted to point out is that we looked at the abundance of different functional groups of algae, and across all different treatments, whether there was lots of algae or very little algae, red fleshy macroalgae were dominant, in particular this one red fleshy macroalga, Melanomantia. And so we'll finish looking at changes in the aerial cover of macroalgae in the corals. To do so, we took pictures of all the plots, again at the beginning of the experiment and at the end of the experiment, and then we're looking at the difference or the change in aerial cover. And so that's looking at difference between initial and final pictures. And we did that by taking these pictures, and then I worked with Clinton Edwards to digitize them. And then we could take the pixels occupied by each of the different kinds of organisms or functional groups, exemplified by the different colors, and convert those into area, knowing the dimensions of the frame. And from there, we can look at changes in the area of these different substrates. So first, we'll walk through changes in macroalgal cover. So first, we saw overall a significant effect of treatment. So there were differences among treatments, no effect of, or there were no differences among the different spatial arrays of those treatments. Let me just show the data here. This is how, let me just walk through this figure first. So we have our different treatments across the x-axis. We have no urchin and open. These are our two controls as identified here. It's a complex plot. We have our 3 single species treatments, Heterocyntrotus, Echinopteryx, Strepnustes, and then our 3 multi-species treatments, Strepnustes, well, these pairwise combinations. And if we compare these treatments with echinoids to the treatment with no urchins, we can infer whether or not they appear to have no grazer effect, right? So if they appear similar to no urchin, that means there's no grazer effect. And any deviation from no grazer effect as we reduce the amount of algae, that's indicative of a stronger grazer effect. And we'll see what the data look like in 1 minute. And overall, we're looking again at the change in macroalgae in terms of the square centimeter coverage on the plot. So these are what those data look like. So when we have no urchin, we have an increase in macroalgal cover. When we have an ambient community, we see no change in macroalgal cover. Interestingly, when we have Heterocyntrotus, we see also a huge increase in macroalgal cover, no different than if there were no consumers present at all. With Echinopteryx, we see a strong effect of consumers. These 2 treatments with Strepnustes are intermediate. And then any treatment with Echinopteryx, ECHM or ECTG, have very strong effects on macroalgae. So again, I'll go through here. Overall, we see big differences among treatments. If we only compare single species to multi-species treatments, we do not see an overall effect of diversity because there's a lot of variability between these 3 and a lot of variability between these 3. And again, blocking is not significant. What this suggests is that this Echinopteryx calamaris, this one species of urchin, is dominant in the system. And this agrees with what we've already shown in that it has some of the highest grazing rates across these 3 different species. So now we can look at the same kinds of dynamics for coral cover. And we'll look at the plot. It's in the same structure, again, with our controls, single species treatments, multi-species treatments, no urchin indicating the effect of no grazers, open, the effect of ambient grazers. And now opposite to macroalgae, as we go up, we're seeing a stronger effect of consumers on increases in coral cover. Overall, we saw strong differences among treatments. When we compare single versus multi-species, we don't see a diversity effect. And these are what the data look like. So generally, when there was no urchin, we saw a slight decrease in the cover of coral. This is where we saw a large increase in macroalgal cover. In open plots, we saw a slight increase, and across all urchin treatments, we saw a slight increase in coral cover. And in one treatment, we saw a very strong effect of the echinoid community on proportional coral growth. And when we compare the positive effect of this one treatment versus the effect of the 2 individual species treatments, we see that this effect is much stronger than the effect of either of these 2 species, indicating that there's an over-yielding or a diversity effect that might be complementary, such that this one community of echinoids results in significantly greater coral growth on the reef. So in conclusion, coral area generally decreased when grazers were excluded and increased in all plots with consumers, but this was more or less highly variable. Heterocyntrotus had very few effects on macroalgal biome or abundance. Echinopteryx was a dominant grazer of macroalgae in the system. And this one community type, this ECTG combination, appeared to have markedly higher effects and improvement in coral cover across versus all of the other treatments, and much higher than any of its constituent species. So in conclusion, these certain echinoid communities may yield significantly greater benefits to these coral reef systems. They do differ in terms of their benthic. So to wrap up, theme 1, we looked at differences in Maui's shallow nearshore coral reefs. And our standard model, or our general model, was examining the effects of consumers, nutrients, and sediments. And we actually collected data to ask how these different dynamics might be impacting corals in the system. And this is my interpretation of what our results suggest. Instead of there being large fishes and echinoids dominate these shallow reef systems. These echinoids, based on experimental manipulations, had strong effects on macroalgae. Nutrients in the system were relatively low and didn't seem to correlate much with development on the tiles. Sediments had strong negative effects on coral, and it appears that sediments actually prevented consumers from reducing filamentous turfs and benefiting these calcified organisms. So just in summary, what I just said, these echinoid-dominated systems exhibited strong effects on macroalgae, nutrients were low, all these sites were sediment stressed, and sediment stress appeared to prevent herbivores from transitioning the system to a calcified state. And this is in agreement with what we term the sediment-suppressed herbivore model, where sediments prevent these herbivores from having the predicted effect based on relative dominance. And so what are some of the conservation implications of this work? Well, one of the important things we need to consider is how do these different sites vary in terms of the processes that are driving their benthic communities. And so what we've shown is that there are 2 sites that have low sedimentation rates and high coral recruitment. And it suggests that if we enact conservation efforts that improve those sites, such as protection of herbivores that would reduce macroalgal growth, we might see a strong conservation effect at these sites. In contrast, we had 4 sites that had very high sediment exposure, very low coral recruitment, and enacting measures like a marine protected area likely won't have much of an effect on coral recruitment. And we had 2 sites that were odd in that they had low sediment exposure, relatively high grazing effects, and still had low coral recruitment. And this might indicate there are other stressors at these sites that we need to be aware of and to think about that might be limiting coral recruitment. So for theme 2, functional diversity of herbivorous echinoids, what we've shown is, like Darwin's finches, these echinoids appear to have developed unique niches or unique ecological behaviors and ecological functions in the system. And again, they differ significantly in their metabolic rates, their grazing rates, their preferences, and their ecological effects. And these results agree with ecological theories such as optimal foraging and niche partitioning. These differences also match taxonomic groupings, indicating that they're evolutionarily significant. For example, the 2 lowest metabolic species were both echinometrids. These 2 individuals that were feeding on Acanthophora, these blooming algae, Acanthophora and ulva, were both diadematids. And then Trypnostes is a toxopnustid, and it's the one species that preferred that chemically defended turbine area. Furthermore, the unique grazing behaviors of Echinopteryx calamaris and Trypnostes appeared to be complementary and good for corals based on our experimental manipulations. In contrast, these 2 species, Heterocyntrotus and Echinometra, do not appear to be particularly good for reef growth, certainly because they don't appear to remove much macroalgae, and according to the literature, they actually remove a lot of carbonate. So the net effect of any algae they do remove may be counterbalanced by the amount of carbonate that they remove from the system. And last, these echinoids exhibit very unique niches, and these differences should and can be explicitly considered in ecological models, surveys, and future management efforts. And so the next chapter that I'm moving on to now is quite a bit different, but it still keeps me immersed in nature, which is an important aspect of my career. And so I'll be moving on to work with Dr. Jim Hobbs in the Fish Ecology Lab at UC Davis, in the Coastal and Marine Science Institute and the Interdisciplinary Center for ICPMS. And we'll be looking at the ecology and conservation of endangered fishes. going back to my fishing roots. And we're looking at longfin smelt and delta smelt that are exhibiting or experiencing a really hard time right now, especially with the California drought, and also looking at steelhead and other associated common fishes all throughout the San Francisco Bay region. And to conclude, I'd like to thank a lot of different people that have supported this work. I want to start off just thinking about my funding, the Ford Foundation, NSF Eigert, Bryven Davis, Scholarship Fund, Scripps Family Foundation, Anna Bailey Sussman, Mia Tegner, other donations from the Sci Fund donors, Laura Fain, the Patty Foundation, San Diego Foundation, and of course the Center for Biodiversity and Conservation. And I've worked at a variety of different, or worked at a variety of different institutions or with different aspects of UCSD, so Scripps, obviously UCSD, the Maui Ocean Center was important, CNBC here, San Diego State, the National Academies, and the Corralling Lab in the Maldives. And of course, I want to thank my advisor, Jen. She's been an amazing guide on this journey and has provided excellent feedback. And of course, the rest of my committee, Andres, Jim, Lisa, John, and Stuart. Mark Miller, who is actually here, and Emily Kelly have been absolutely fundamental and foundational for me being able to begin and continue my work in Maui, and I cannot thank them enough for all that they have done. Don McLeish, I can't explain how beneficial he has been. He's a self-made engineer, and he helped me design many of the experiments in Maui, just out of the goodness of his heart. And the sponge crabs for you, Don. It's another story. There's a huge Maui family. Again, there's no marine lab in Maui, and so all of this work was out of minivans and in people's kitchens, oftentimes. And I just can't thank this support team enough. Again, Maui Ocean Center, Division of Aquatic Resources, Mark, Darla, and the various members of Makai Watch, Donna, Karen, and there are many others, Rich. I've had an insane number of field and laboratory assistants, as Jen has already mentioned, and I'm just going to thank all of you right now, because there are just so many of you. Clearly, the Smith and Sandin labs have been phenomenal. Clint and Jackie helped me start the experiment. And again, there are just so many, obviously Penny at CNBC, Gideon, I can't thank all of you enough. I've had also a much larger family at Scripps that have been absolutely phenomenal. My 2009 cohort, the group in Tides and G-Dog, you guys have been a huge support system. All of the different sports teams, Maniacs and the Ball Bouncers, everybody in the grad office, Gilbert and the Maureen's and many others. Crystal, who's here, she was there at the beginning. Josh, who's here, he was there at the beginning. Often serving multiple roles. And of course, the dive office, Christian and Rich, have been absolutely amazing with respect to supporting all of our diving activities. And we spend a lot of time diving underwater. I need to acknowledge Adi and her website at Delesia. She created all of the graphic art that you've seen in this talk. And she's now a professional scientific illustrator, getting paid for her work. And she's also been published in several different manuscripts now. And so I highly encourage people to seek her out and to use her skills. And I've had various interesting encounters. Ned Deloach, who's a phenomenal naturalist, and Burgundy's helped me get through very long days in the field. The family, the Alatis and the Maldives and Northwest Islands crew, and my favorite boat captain, Calais. And then of course, I can't thank my family enough for always, an extended family and friends, and they've always inspired me to, again, be in nature and to think about nature and to pursue greatness in whatever I pursue. And then my own personal Wonder Woman, which I'm looking forward to spending the rest of my life with. And of course, I'm sure there are many people I've missed, and just come talk to me personally and I'll thank you. I'll shower all kinds of things. And with that, I'll just thank all of you for being here and for listening. Thank you, Shukriya and Devehi from Maldivian, and Mahalo Nui Loa from Hawaii. And with that, I'll be happy to take questions. Yes.
What would happen if, like, just through I don't know, just all kinds of, if 50% of our reefs would disappear, what would happen?
Well, we've actually seen that happen in many different places. It depends. Many different things can happen. For local, many low-lying island communities, for example, on atolls all throughout the Central Pacific in particular, are dependent upon their reefs for a variety of different resources. It could be food, it could be tourism, or just shoreline protection. So there are many different aspects of that loss that can occur over different time frames, right? So if it's tourism, if you lose a chunk of your reef and now it's ugly, people stop coming and you lose a huge chunk of your economy. If you're dependent upon that reef for food, that reef dies off. Ultimately, you lose the three-dimensional structure, you lose the production of fish, you lose your food source. And through even longer periods of time, that reef will erode away, and suddenly now you've lost your barrier to big waves, and those waves will now start passing through this eroded reef and will start eroding your island. So in particular for island communities, those effects can be really severe, both over the short term and the long term. To me in San Diego, it doesn't do anything specifically, but just knowing how beautiful and amazing these reefs are and the fact that they hold so much biodiversity, much of which is unexplored. Also, we keep finding new drugs. In fact, there's several groups at Scripps that are currently exploring coral reefs and developing new drugs for treatment of cancer and a variety of other things. And so as we lose these reefs without even knowing what's in them, we don't even know what we're actually losing. And we're impoverishing ourselves in ways that we may not even be able to grasp. Yes?
So I was struck by the results you had for the coral, or the algal biomass, and the effective version of algal biomass versus the coral cover.
Yes.
Especially seeing the algae go up so much when there's two species, yet at the same time the coral still goes up.
Yes.
So I had a question about what you thought there's more going on to this idea of like algae up, coral down, or it's more complicated, or if this is something experimental.
The simple answer is it's more complicated. Some corals are really good at competing against algae. Porites is well known for being able to hold space, even with pretty large macroalgal blooms. And if those blooms are controlled in a reasonable amount of time, those corals can then regrow. Other corals aren't able to hold space in that same way. So what we oftentimes see, and there's kind of a large debate in coral reef ecology, is chicken or the egg. Do we see algae increasing on reefs because coral died and the algae is recruiting to that empty space? Or do we see corals dying because algae are growing out of control? And my results suggest that these corals are actually pretty good, at least at these sites, in holding space. And the algae are only really able to have massive effects, at least over a 2-year time period. This is a relatively short experience given the lifetime of a coral. But over a 2-year time period, these corals are actually pretty effective at holding space. Much of the increase in macroalgal biomass and cover is on areas that didn't have coral at the beginning. And so they're turf communities that have now developed into fully grown macroalgal communities. I think, does that answer your question? Yeah. Question?
In fisheries, we think that recruitment is often related to how many fish there are. So you look at how it affects coral recruitment, but how much does the amount of coral spawn affect coral?
That's a great question, and it's clearly really important. For example, in the Caribbean, with the large die-off of diadema and the dramatic basin-wide changes in coral cover, they've had many decades of recruitment failure. In Maui, there's definitely variability in the abundance of adults across those different sites, as I showed you in coral cover. In particular, one site, Olowalu, has been mapped, and it has the largest intact reef system in the entire island, maybe even in Hawaii. And so at that site, we would expect there to be a lot of coral recruits. But similar to fish, these organisms are broadcast spawning their eggs up into the water column, and those can get transported all over. And some researchers have modeled, Kurt Sterlazzi and some others, have modeled where those recruits go. And whether or not a given site is a source or a sink is still to be determined. And I really don't know for some of my sites. Olowalu is definitely a source for many different parts of Hawaii, certainly the Maui Nui island conglomerate. But I don't have a good answer for whether or not certain sites are sources or sinks. In theory, a site without a lot of coral could still get a lot of coral recruits from another site. But I don't have all those dynamics nailed. So we don't know the differences in larval supply versus like post-recruitment mortality dynamics. Yeah.
To get into more diet implications,
Yeah.
if Olowalu is currently a less sedimentary place with lots of nice corals, is the plant development development still on, or did they cancel that?
It's in limbo right now.
Yeah, it's being fought. Yeah, so there are a lot of efforts to develop the island of Maui for economic purposes, and many different groups are fighting some of those developments for exactly those reasons. Question up in the back.
Thank you, Levi. This was a great presentation.
Thank you.
I'm just wondering about the sediment stress sites. Given that you have some sites that have low coral growth and low sediment, what makes you think that it's actually sediment that is limiting the sites that have sediment stress? Could it be the same other factor?
It could be. I can't say for certain it's not, because again, that's just a correlation. But there was a very strong exponential relationship, right, between sediment accumulation rates and coral recruitment across all the different sites, both in and outside of cages, indicating that it's probably not a macroalgae thing, right? There are several other sources of information suggesting that each site might be very unique. And that's why you might see some sites with low sediment still not having high coral recruitment. It could go back to Brian's question of larval supply. We don't know what supply is to these different sites, so maybe that's driving those different patterns. But there are other things. So for example, at Kaikili, it's the most used beach area in the entire island. And there's been a recent series of publications indicating that oxybenzone in sunscreens is extremely toxic at very low concentrations. So maybe there's some effect of sunscreens at that one site. Also, that site has huge wastewater inputs that have a variety of problems. And this is something Jen has actually been working on for a long time. So maybe wastewater is limiting recruitment at that site. In contrast, La Perouse on the other side, the far southern end, had low sediment and low coral recruitment, but it had huge numbers of Echinometromathii, one of those urchins. In fact, it had that 600 grams per meter square. And research from Kenya by O'Leary and McClanahan and others have demonstrated at those high densities, that specific species of echinoid actually might start eating corals. So each site might actually in some way be so unique that the general patterns may not be really clear and obvious. And so I think our study, and this is what the nice thing about thinking about the conservation implications, our study showed that those two sites were anomalous in terms of sediment exposure. And so additional studies exploring these different factors are probably warranted at those two specific sites. Yeah.
At Kaikili, has there been a study to, because that's the most protected fish right now for fishing and stuff like that. Has there been a study around? Has there been a study to see if the increase in fish herbivore has decreased the turf at that site yet?
Yeah, so they've been monitoring for a while, and they found that CCA appears to be increasing. And CCA are those crustose colorant algae that are also calcifiers similar to coral. But it's probably going to take more time for herbivore biomass to build up to the point at which we see really strong differences. And that's something that Emily Kelly is continuing to work on with the Division of Aquatic Resources. Yeah. Question?
Great job.
Thank you.
This is kind of like the same line in the fish.
Yeah.
Places that, you know, a lot of these haven't really changed since you can monitor.
Right.
Limits of learning places, maybe some of the effect of economic were emphasized because it's a diverse species.
Yes, that's a great question. So I'll see you on one side, and then I'll double down on another. So yes, Emily and I actually we're working on a paper together right now where I've contributed my grazing rates from my grazing assays into her algal consumption budget for the reef at Kaikili, and that's something that we're currently working on. Also, we've been thinking about shallow versus deep. So these really shallow reefs, they're in 2 to 3 meters of water. And in Kenya, they've shown very similar patterns where we have really high echinoid abundance, really low fish biomass. And as you get deeper, you start seeing much higher fish biomass and much lower echinoid biomass. One of the reasons is predators. So in the deeper reefs, you have really high abundances of predators, and the predators are actually able to regulate echinoid abundance. So it may not be an herbivore, herbivorous fish issue, it may be actually a predatory fish issue. And I've started looking at some correlations between echinoids and predators, and I need to go into more detail about that. But in Kenya, they've shown very strong patterns between a single triggerfish, the red line triggerfish, and Echinometromathii abundance. And it's exactly what you say. The effects of fishes, herbivorous fishes versus herbivorous echinoids changes simply due to the presence or absence of that one predator. And so there are lots of different dynamics like that that we can think about. Yeah, thank you.
So you said you went
Say one more time, sorry.
you said you were
I did.
So
Decoloration. Yeah. Yeah, so bleaching is a massive problem. So when we have anomalously warm years, the water gets warm, the corals spit out their, I didn't go into the physiological details of corals, they spit out their zooxanthellae, which are little algal symbionts, which are important for the corals to be able to feed themselves, basically. And so in certain years, especially El Nino years, we see really kind of global events of bleaching where reefs all across the globe spit out those zooxanthellae and the corals die. And the Maldives and the Indian Ocean, I'm not sure specifically why, but in the Indian Ocean, it seems like a lot of those effects are really strong. And they've had massive bleaching. So those kinds of acute disturbances might overshadow any of these other kinds of disturbances, whether it's grazes or sediments or anything, if you're having 50% mortality in a single summer due to bleaching. And in some places, we've seen that. The Great Barrier Reef just recently had one of those mass bleaching events. Hawaii historically hasn't had those kinds of mass bleaching events because it's a high latitude reef area, but just last year and the year before, it had one of the first and the worst bleaching events. And whether or not that's going to increase is yet to be seen. And if it does, it could negate some of the effects of consumers. Though at the same time, with those kinds of acute stressors, it becomes even more important to have those grazers to keep the algae crop down so that once those stressors are removed, once conditions calm down or cool down, the corals can start to recruit again, and they're not outcompeted by these huge blooms of fleshy algae. So those are definitely important. I don't know if I answered your question, but those are important things to consider. And those acute stressors and how they interact with some of these more chronic processes is an important field that people are studying. Yes?
So I'm wondering whether you notice any difference in male activity between the expectations of the tested. Enclosures reduce pressure of light.
Yes.
I think we've talked about this before.
Yeah.
But your enclosures reduce the light level probably quite a bit. So I was wondering whether you think the trend, metabolic trend would stay the same if you tested them with enclosures.
Yeah, that's a great question. So all of the metabolic rates were measured in the dark because we didn't want our activity above scaring the urchins or making them behave unnaturally. So those were all dark, and we wanted them to just be calm and resting. Certainly with light stimulation, that could change, and we didn't test that explicitly. We just wanted to have a standard measure to compare species. So that's something that we should probably do in the future. That would be really interesting. The question about activity in general on the reef, I believe Emily and I did some nighttime versus daytime surveys and didn't see huge differences in densities in terms of what was out on the reef. But 3 of those species, the high metabolic species, the 2 echinopteryx, the spines, and the collector urchin, all have chemical defenses. They have venoms. So they're actually able to roam around the reef freely during the day. And for the most part, they don't get attacked by predators. Whereas the 2 echinometrids, the pencil urchin and the smaller rock-boring urchin, are highly susceptible to predation. And so they bore into the rock and they tend to kind of hide out, which again feeds into the fact that they have low metabolic rates. They don't need to search out lots of food, so they can kind of just stay hunkered down. And they'll eat whatever is available because they can't go out and search for better food. So those guys stay hunkered down. And in our surveys, they stay hunkered down day or night. In other places, though, where there are no predators, those guys can actually leave their holes And you will see daytime versus nighttime differences in terms of their activity. So it depends on the species, it depends where you are, and it depends on some other processes like predation and how these organisms behave. I believe on the reefs that we were looking at, for the most part, all the echinoids were active during the day. The reason we put them in those shaded enclosures is because we did assays on urchins that were fully exposed to light, and it worked fantastic for the high metabolic species that are already out during daylight. When we tried to do the metabolic assays with those that bore into the reef, they freaked out and wouldn't eat. And so we decided to put all of them in a protected space, just so that we could compare grazing rates among species.
I think we're going to have to cut questions off at this point. But again, I'd like to thank everybody, thank his committee and all of his friends, collaborators, and general support team for being here. I'd like to invite the committee to stay back and we'll continue asking Levi questions. And the rest of you, we hope to be celebrating at Surfside around 2 o'clock.
Yeah, and you can head over there early. People will be setting up. Start eating without me, that's fine. And I'll be there as soon as the committee's done with me.
Thanks again.
That's fun. Okay. It's yeah. And anybody that's watching, thank you for tuning in. I look forward to chatting with you and all of your comments.
Good job. You did great.
Yes, what's going on?
I'm being a little. What did I do?
Barely said hi.
I'm sorry.
Looking
I'm sorry.
down and
I'm bad at eye contact. You know this.
I know that. Like,